Thank you everyone for coming and for everyone who's joining online as well. The idea of these workshops is really to equip people with practical skills for image analysis. And so you don't necessarily have to attend every one, but we'll sort of work through in this order. And normally we start with just sort of how to use ImageJ and then onto more advanced things. But I think for many people that I've met here at OSU, working with Ilastik and doing pixel classification is something that will immediately have great benefit to their work. And so I thought we'd just kickstart this whole thing, learning how to do machine learning image segmentation, and then go into some more advanced deep learning restoration and segmentation techniques, and then finally come back to how to automate these things using ImageJ, which is a preference for some people to use. And like really the goal of all of these talks is and workshops is to give you practical skills. So not just talking about theory, we really have practical skills and tools that you can then take away immediately and use for your research. I just want to add, I've made a 2 hours time slot for these workshops, but it doesn't necessarily mean that we're going to be working on this for 2 hours. Some of them may be an hour to an hour and a half, but I'll be available for the full 2 hours. So if you want to ask questions afterwards, or we just carry on the workshop on specific things for your work, then please feel free to do that and to ask questions. And so the main reason we're talking about these advanced tools is because when we come to do biomedical image analysis, we have this idea, maybe especially when we're first starting out, but even when we've been away from it for a while and we come back to it, we have this idea of, okay, I'm going to analyze some organelles or maybe some cells. We sort of think maybe it'll be easier than what it is, but we start to do the imaging and immediately we realize just how complicated it can be. Like biology is really messy, and the more you look into it, the more everything is just connected. And so we have this simple question of, okay, I want to quantify the number of endosomes, or I want to measure volume. And then we start to see, okay, but if I change the brightness and contrast a little bit more, suddenly there's 10% more endosomes, or they're connected to these other endosomes. And how can you really quantify that and report that? Because those sort of measurements can have a meaningful difference in how your study goes and what you can actually extract from these images. And it becomes even more complicated when we start to work in tissue, especially three-dimensional tissue, because again, this idea of neatly partitioned cells just doesn't stand up. You look at the tissue and things are just very complex, overlapping, intermingled, and using standard approaches to biomedical image segmentation will just leave you very frustrated. And I think a lot of people, especially over the course of my career, working with people, if they don't have these tools, they just revert back to doing things manually. And that's something that we want to avoid. And for many reasons. One, it's time consuming. It takes a lot of your time to be doing this manual work. It's a big opportunity cost for you. You could spend a huge amount of your project just manually clicking on things when you could be doing other more important work. I mean, all of you are very intelligent people. You could have much more impact on the world if you weren't just doing these mundane tasks. And so that's one part of this. But I think a more important aspect is when you're doing this, you're getting limited information. Like if we just clicked on all the cells here, we get a number, but we're missing things like the orientation, the pattern of labeling inside those cells, the area or volume. So we're really cutting down the information we can get out. And then it's highly variable. Even if you yourself have very strict rules about how you're planning to count these cells, day to day, it's going to vary. It could be the intensity was slightly different the next day, or you were in a room that was slightly brighter and the screen was more difficult to see. There's so many things that can impact that, as well as the bias. Even if you're not blinded, and hopefully you are, but if you're not blinded, then you may start to skew the results in one way or another, even unconsciously. The last point I wanted to make was that this inter-observer variability too. So if you just look within your own lab and get different people to count the same image, you'll find that, especially if the image is complex, there'll be a lot of variability between people. And this is a problem too. If someone else wants to reproduce your research, a different lab is going to have even more potential variability. So what we want is something that is a readout that provides insights into the biology and reflects the biology accurately, but also has these other characteristics. It should be automated. And when I talk with people about this type of work, the first thing they think of is, oh, great, this is just going to save me a lot of time. But I think this is probably one of the least important aspects of what it allows. It allows you to do repeated measures. So in 10 years' time, you can give this code to someone in the lab, they'll get the same results if given the same image. And I think this is really important, especially in the situation we're in where we have a reproducibility crisis in science. So creating tools where you can actually openly share what you're doing and have someone else reproduce it is incredibly important. It's rigorous, it's very thorough, so we can get all the information that we are sort of targeting to get from the image, not just maybe the number of cells. And it's also can be robust. So you can make an image analysis pipeline that will work really well for maybe one image, but as soon as you go to another area in the tissue, this pipeline will fail. And so machine learning and deep learning tools allow us to have something that's more robust and work well across different areas and different tissue. And as we mentioned, it should be unbiased, so we can get measurements that aren't skewed by what we want to see. And I think finally something that's important to the work that I'm doing, and hopefully to you, is that we're creating tools that are open. So when we're making a tool, if we can do it without having to use commercial software, it means that a lab somewhere that has different resources or different budgets can still do this work. And I think that improves science for everyone, as well as letting people interrogate what we've done. The way many people think about image analysis is like this. They have the imaging and then they do some analysis, maybe in Imaris or ImageJ. But in reality, this image analysis pipeline is far more complex, especially in the last few years when we have started to add more tools. So not only do we have more sophisticated ways of dealing with images, so image restoration, which we'll focus on in this series, better ways of segmenting, how to actually deal with the statistics and plotting of this data in an open way, but also on the further to the left, which I think is an area that requires as much attention because it's much more easy to solve problems to the left of this sort of flow. So, going all the way back to fixation, the way you're mounting the slides, the way that you're imaging, those things will have a much more bigger impact in improving your results than trying to tweak things at the end on adjusting segmentation algorithms and restoration. And so even though the series is really focused on this image analysis and processing, I thought because this is the first session, we could go through briefly and talk about things that can help improve your outcomes in these earlier parts of the pipeline. And I'm not going to touch on fixation, but I think when you start to think about how it's easier to solve problems earlier on, it even makes you think, okay, is PFA the right choice for my fixation? That's what people have been doing for so long, but now there's a lot of work starting to show that maybe glyoxal is a tool, a solution that can actually fix better. So even the way you fix the sample can have an impact on the final result. Okay. And so just some points that I think are really important and areas where I've seen labs struggling in the past. People can have the best intentions and really be well educated on how they're doing their imaging and their analysis, but they can miss these simple things. And I think the first one, which I still catch labs with, is making sure they're using the right cover slips. So all microscope objectives are designed to work with cover slips that are 0.17 millimeters thick. So this number 1.5 thick cover slips. So if your lab is using number 1 cover slips, which is very common, that's too thin, and vice versa if they're a larger thickness. And the biggest difference is, if you're just doing very simple imaging with the 10 times objective, you won't necessarily notice these differences. But as soon as you start to do high resolution imaging with oil objectives, you can see in the plot on the right-hand side, you'll have a drop in intensity. So as well as other effects coming into the imaging. So it's really important just to solve this simple problem by using the right cover slips. I mean, I've mentioned that it's not always the case. So there are situations where you might be working with difficult thick sample using an oil objective, and you may want to use a thin cover slip, and you're mounting in something that has the same refractive index as glass. Ordinarily, especially for most projects, just using the right cover slip is the way to go. And some other, another sort of very simple thing, but I think it's important, is thinking about how you're mounting the slide on the stage. So if you're using an inverted microscope and you're mounting the cover slip across the full length of the slide, that will actually introduce an angle, and then you'll start to have aberrations in your images. So try to make sure that the slide is flat. And that even comes down to if you're labeling with tape. If you just put the tape on the slide, that's already adding an angle too. So simple things, but can have a big impact on the quality of your work. But the other thing that really comes up a lot is labeling and seeing a lot of aggregates in the tissue or in the cells related to antibodies. And I think a very simple thing that people can do, which they may not be aware of, is just spinning down antibodies and filtering them before use, especially some problematic antibodies. You see this a lot with like some of the secondary probes. If they're a bit messy, then you'll see these sort of floating aggregates in the tissue. And so I think this is something that's very simple to do that can improve your chances of success. How you're mounting the samples, what you're mounting them in. So I still encounter people using mounting media with DAPI in it, which is like you think it's a good time-saving technique, but you get much worse results in terms of the contrast and labeling of the nucleus. And you have this background of unbound DAPI in the media, which creates a background during your imaging. So definitely skip that. Just use good mounting media and just perform the DAPI labeling step separately. It's very quick and it works well. And this is a slide from a while ago. I'm not trying to push people in the direction of using Vector Shield. In fact, Vector Shield is known to quench things like Alexa 488 and Alexa 647. So if you're using Vector Shield and you're having unsatisfactory results, that's a good reason to switch. But just showing you that different mounting media can have different effects on the tissue. So a hard setting mounting media can really start to shrink down the tissue or the cells. And so if you're trying to measure the three-dimensional volume of cells, maybe you don't want to use a hard setting mounting media because that's really going to shrink down the z-axis of the tissue. And likewise, the refractive index of the mounting media is important too. So whenever you have this mismatch between the mounting media and the solution that you're using on the objective, so say you've got a water objective, but you have a very high refractive index mounting media, or vice versa, you have oil objective, and you have a normal tissue section is in a sort of aqueous mounting media, then when you're imaging, you'll have great results near the cover slip. But as you go deeper, you're losing signal and contrast. And you probably think, oh, it's just because I'm going deep into the tissue. But in fact, it's just this refractive index mismatch. So solving this can make a really big difference. And that, this is a good example of prolonged glass, which is a mounting media that's refractive index matched to glass. So you're going from oil to glass, sort of equivalent to glass, and you can image much deeper. So there are things to think about in terms of mounting your sample to get much better images, much brighter images, better resolution, very easily just by making those changes. Another simple thing, just to use probes that are spectrally separate. So this is something that you can easily check for yourself. I think there's many good tools. FP-Base is one, Thermo Fisher SpectraViewer is another. You can go online and then just see, like, okay, with the probes I'm planning to use or the fluorophores I'm planning to express in my tissue or samples, how separate are they? Like, good example is sometimes people have kiwi tomato and M cherry together, and that's necessarily not going to be very easy to unmix them. There are ways of having the right microscope to do that, but it's better just to choose probes that are spectrally separate. And this is a good way of just checking that before you go out and start labeling your cells. Also, just when you're imaging, I think it's sort of the intuitive thing to do is to start from blue and go to red, because that's just how we sort of think about these things. But in fact, going the other way is a lot better, because when you're imaging with the 405 laser or the UV light, you're actually affecting the tissue as well as the other probes in the sample. So if you have a long sort of exposure with that light, you may end up bleaching out your far red signal before you even get to it, especially if you're doing like thick tissue samples. But and sort of related to this, you also want to think about what you're using these probes for labeling. So if you're labeling something that's very fine, like say in this case, microtubules, maybe it's best to actually use a shorter wavelength probe, because then you'll have better resolution when you're imaging. I think people forget, people know that shorter wavelengths that can give you higher resolution, but the impact is actually quite significant. You can see here, just comparing Alexa Fluor 488 to 647, if you're trying to quantify what's happening on those the microtubules, you have a much harder time if you use the far red probe. And so maybe something where you don't need resolution, like just a nuclear stain, it's better to put that in the far red wavelength, and where you don't have to worry about that resolution. Okay, so that's just all the sort of sampling things, just simple things that I think can have a big impact. When it comes to imaging, we've sort of talked about it already, avoiding this refractive index mismatch, because that's where you have spherical aberration, and that leads to this loss of contrast, loss of signal, and just general problems with imaging. But I also wanted to point out, this isn't going to be a big seminar on imaging. Maybe we'll organize something like that later in the year. But just to let you reflect on, with objectives, there's two numbers that are typically important: the magnification and the numerical aperture. And everyone sort of thinks about the magnification first, but numerical aperture is what really determines the brightness and the resolution of the image that you get. And so just on the far right-hand side, you can see the same magnification with different numerical apertures. You're actually completely losing structural information with that lower numerical aperture. So think about the numerical aperture of the objective that you're using, depending on what you're trying to image and quantify. And then this is actually, the one on the left is sort of more talking about refractive index mismatches and how it can distort the image that you're seeing. So we all think that, the image that we're getting is a rough approximation of what's in the sample, but having these refractive index mismatches really distorts the image that you're getting back. And so structure. So you can have elongated or sort of contracted structures based on this mismatch. I think this is something to also be aware of if we're trying to measure the volume of these structures in the sample. Just another simple thing, I think most of us are using microscopes that are in core facilities, or maybe they're shared instruments. And I think all of us know how much can happen to these shared instruments from one day to another. And you may think, oh, well, that's the responsibility of the platform manager or whoever's sort of looking after that instrument, but they may not be aware of the issue until days later. And so if you really care about the work that you're doing, I think it's good that when you go to the microscope, you have some ideas in mind about what you're expecting to see. So if many of you will be imaging samples over multiple days that are very similar to each other. So you can have in mind, like, okay, how bright is my sample supposedly going to be? Maybe I'm expecting to have counts of about 10,000 gray levels for the green channel. Or maybe even you have an image of what the sample should look like. Because as soon as someone, maybe puts a little bit of oil on the 20 times objective because they're a little bit lazy when they lost where they were when they're on the oil objective, they go back to 20x, they don't clean it. It's very common for these sort of things to happen. The image you get when you turn up at the microscope, you'll still see something, but it will be kind of a little bit blurry. And if you're not used to doing imaging, you won't recognize that difference. And so just having an image to compare to or having some ideas of intensities can save you a lot of trouble. Other things people do is have dye-filled chambers that they know are a specific intensity, or a plastic chroma slide that's a specific intensity. But just something to think about, rather than just turning up at the microscope and spending 6 hours imaging and then later on finding out that all these images are totally different to the rest of the data set. Yeah, so we talked about this a little bit already. Just in terms of saving files, I generally think it's good to save in the proprietary format, because if you're at the microscope and you do these conversions to TIFF, it's very easy to make mistakes in that export process and have adjusted range of intensities or not have the full information. And so just keep them in the proprietary format. And then later on, it's very easy to convert to TIFF or HDF5 or another format that you're working in. And I think it's a good idea. Maybe you're already doing this, but I see a lot of times people will just have images with, very ambiguous names. And if you're planning to do big research projects where you'll be doing automated quantification, if you can put that information in the file name, when you're running your scripts, all that information can be pulled out. So instead of you later on having to go back and sort of correlate, okay, image 5, that was these cells, you can have you can generate CSV files or table files with related information already populated. And this is a big time saver if you're doing analysis projects. Okay, so that's just some tips. I hope that that's not all information to you, and I hope that it sort of can help improve the work that you're doing. So now I really want to get into the idea of these workshops and why we're doing the sort of analysis that we're doing using machine learning and deep learning. When it comes to image analysis and you have images like these where you have very discrete cells or nuclei, it's very easy to solve that problem using automated analysis. And maybe we'll do some problems like this when we look at automation of imaging down the line in the ImageJ workshops. But for many of us, our problem is more complex. We could be looking in cells where there's a lot of varying background and different morphologies of objects. We could be looking in tissue, like this example of neurons. And when we try to apply filters that will pull out those cell bodies, if we just use the same traditional approaches, we'll run into problems. And sure, there's things we could do here to get a better result of detecting those cells. But then when we have another brain section, it's probably going to start to have issues. And so we want something, as we were talking about before, something that's robust, something that we can get working on a couple of brain sections and then give it 100 more brain sections and trust the numbers that we're getting back. And so this is a sort of result that we can get if we start to use pixel classification. And this is where we get a lot more information about each individual pixel and try to classify it as a particular class. So as a cell body or as a dendrite or as background or whatever your particular image analysis problem is. And that's what we'll work on today using a tool called Ilastik. And this is a very easy to use tool. It's not going to solve all of your problems, but it will solve many challenging segmentation problems. And I think the good thing about it is it's very easy to use. So we can get going very quickly using this, and we can also use the results from this to start to train deep learning networks. And so I think it's just a very good foundational tool for this type of series. And so using this tool, we can not only segment very challenging images like EM images, but we can also classify the objects that we're detecting. So in this case, they're classifying nuclei based on a different cell state. And so this is something we can look at today as well. And then just to give you a preview of some of the things we'll be looking at in the subsequent series, more deep learning approaches to image restoration and segmentation. And I think these are just critical tools for research now, because many of you may be working on live imaging projects where you need to image quickly, or you don't want to damage a cell or affect how the normal biological processes in that cell. Or maybe you're on an older confocal and you want to image a large amount of tissue, and it's just not possible without getting very noisy images. And so image restoration provides a solution for those kinds of problems. We can train a network to deal with really just incredibly poor images. You can see on the left-hand side just how noisy these images can be, and get images that very high contrast that we can then segment much more easily. And so I think this is something that, it just really opens up a lot of doors to what's possible to us. And so I think this is a critical tool. And just some more examples of how you can use these tools. And you shouldn't just think about it in terms of restoring signal and contrast. These tools can also be used as in kind of an upsampling way to make data sets more isotropic. And so we can, this problem we always have on imaging on a confocal is we have this poor axial resolution, and it's very hard sometimes to see things in a true 3D structure. But we can train networks to help us get closer to isotropic data. And this is really useful for, again, volume measurements or segmenting cells. And so we'll look at this. And then just sort of trying to expand your mind in terms of what's possible using these types of approaches. Restoration can also let us, so this is Arken is another deep learning network that allows image restoration. And I think this is a fantastic paper from Hari Shroff's lab, which I recommend if you're interested in this to have a look at this paper, because I think they sort of pushed it to the next level by actually going beyond the resolution of the microscope. So in this case, they expanded a sample. So using expansion microscopy, they expanded a sample and then trained a neural network so that they could image on the microscope and try to restore images as close as they could to an expanded sample, even if the sample hadn't been expanded. And I think it's very surprising to see these results. And I think they, obviously we're surprised too, but these are the sort of improvements they could get in terms of resolution by using this approach, just by, giving a target of this expanded tissue and trying to restore normal images to that scale. And so these are sort of things that you can think about if you have a project where you're really trying to get to the bottom of complex structures and you need more resolution and you may not be able to go to EM or expand the tissue. And the same applies to other dimensions as well. So there are some, there's a very interesting paper that came out recently looking at how to deal with the problem of when you're imaging over time, especially if you're doing 4-dimensional imaging, you have gaps between the volumes and objects can move quite a reasonable distance. And so how can you actually fill in those frames so you can track those objects better? And then the sort of following on from that, we'll get into segmentation and the right tools for the right job. So whether it's just being able to segment in a way that's better than using Ilastik, or deal with like very tightly clustered nuclei, as you may find in very dense cell tissues where nuclei are really just are touching each other. It's very hard to separate them. But also in other types of cells where you have, like the example I gave at the beginning, where all the cells are really touching, how do you separate those cells reliably so that you can actually measure their volume? And so we'll look at some tools for doing that. Okay, so as I said at the beginning, this is really focusing on the machine learning using Ilastik, and in a second I'll just get set up so that we can go through that together. And then the subsequent sessions will be focusing on this sort of deep learning approaches and using ImageJ.